Hey everybody, I'm Chavi, and I am a graduate student in the UW Genome Sciences department. And today I want to talk about a paper I've been reading by Hussein Al-Asadi, studying the idea of demographic modeling and the tool that they have developed. So the first point I wanted to make was that geographic information with genetic data can be a really powerful resource that can shed light on really cool and important phenomena like migration, evolution, and conservation. The first example I wanted to make was this idea being applied to humans, where geographic sampling data was taken with genetic variation to develop this theory of the out of Africa model, which has been largely accepted. The idea being that humans originated in Africa and we have since spread out to the rest of the world. And the second example I wanted to give was another cool focus on conservation, where a group observed that these bighorn sheep of California, the populations are completely segregated by human-created barriers such as roads and walls. And that has a really big impact on how the population mixes, which can make the species itself more vulnerable. The paper I'm talking about today is a paper on demographic modeling, which is an initiative to try and represent a population that's being observed with a mathematical model. So the two variables that it tries to represent and it tries to make estimates for a given population are migration, or how different populations are mixing with one another, as well as effective population size, which is a really common statistic used in population genetics and is a way to represent the variation of a population and the health of a population. And this is a really interesting initiative because it can tell us about how we are changing over time, and it can shed light on really interesting events in history, but it's also very medically relevant. So here I'm showing one of the Manhattan plots generated in a GWAS experiment, which is an initiative to look at lots of sequencing data and to find candidate causal variants associated with disease. And these are initiatives that have really driven lots of biomedical research, really productive biomedical research, but in some cases they have led to red herrings. And that's because we can find associations that seem to cause certain diseases when they can actually be explained by drift. And that's something that needs to be tested for with strong demographic modeling. So the tool I'm talking about today is called MAPS, which stands for Migration and Population Size Surfaces. And it's a tool that tries to make estimates of demographic models over different time points in history, which to me is really cool. So I'm going to walk you through how it works and going to give you some of their proof of concept simulations, and then show how they applied it to model European migration history over the last 2,500 years. So the first step of this tool is to gather input data. And what's needed is demographic information of samples, which can be thought of as the sample's sort of geographic origin and heritage, as well as matched genetic data, which can come in the form of either whole genome sequence or SNP microarrays. And what comes next is genotyping that genomic data. And what that means is to basically go across a given reference genome and determine the two alleles that a sample has over given polymorphic sites, as shown in this example image here. And with genotyped data, the next step is to phase the genotype data into the maternal and paternal haplotypes of a given sample, and then to determine common haplotypes between the individuals. And with those haplotypes determined, we identify and compare haplotypes across the populations and determine identity by descent tracks, which they call these long pairwise shared coalescents, but which are commonly referred to as these IBD tracks. And what these tracks are stretches of DNA with shared alleles across the entire stretch between some haplotype A and haplotype B. And that sort of those shared alleles can be explained as a being sort of co-inherited from a common ancestor. And because of recombination, we know that these IBD tracks decay over time. So a longer stretch of IBD sequence can be thought of as evidence that these two individuals have a haplotype with a more recent common ancestor, as shown in this image on the bottom. So after sort of identifying these IBD tracks between all of the pairs of haplotypes we have, we then generate a table and essentially count the number of IBD tracks over given ranges. So in this example, I'm showing this toy sort of table comparing haplotype A and haplotype B, and they have 10 IBD tracks between 2 and 6 centimorgans, 3 between 6 and 8, and 2 that are greater than 8 centimorgans. And centimorgans are units of genomic length. And they have a lot to do with recombination, but for our purposes, we can think of them as just a stretch of DNA that on average in the human species is around 1 million bases. So in the end, we sort of count these IBD tracks of different ranges between different haplotypes. Then we take that table we generated and we push it out to a subpopulation level. So we compared 2 different subpopulations and we find all of the haplotypes in subpopulation A and subpopulation B, and we count all of the IBD tracks we find over a particular range. So here I'm showing some illustration of a heat matrix where, 3 is sort of the more counts of particular IBD tracks and 0 would be less. And we do this for each sort of range of IBD tracks, which can be thought of as the different time periods when different haplotypes have shared common ancestors. Then with this information, these different similarity matrices over different IBD track lengths, and using this stepping stone model to basically relate different subpopulations over geographic distance, MAPS generates estimates for migration and population size over different time periods using a Markov chain Monte Carlo process. And I'm not going to get into the math right now. That will come in a later video. But for now, I think it's worth mentioning that they start with some prior using a genetic map and looking at genetic similarity between individuals. And then they just basically iteratively change their population and migration parameters for each subpopulation to come up with a model that best explains the relation they find in their data. So how well does this tool work is the next question. And the developers wanted to first test this demographic modeling tool using simulations. So they used MAPS, which is a coalescent simulation model, a coalescent simulation tool that can produce present day genomic information given some user-defined population parameters and migration parameters and historical events. So they basically created this simulated world of 20 subpopulations where each subpopulation has as a baseline 10,000 diploid individuals and a migration rate between neighboring subpopulations of 0.01. So one person migrating between regions on average. So they start with this baseline, and then in different simulations, they introduce some event that changes suddenly the migration or the population size. So in this example here, I am showing that a situation where at around 10 generations, they have this sudden decrease in migration. And that can be a result of a geographical change like a river or something else that has caused a sudden drop in migration. And then they basically took this data and tested out their MAPS demographic modeling tool with it, and they wanted to see how well their tool recapitulates this sort of user-defined history. So they started with 2 simulations where they changed migration. The first was this one where I just showed where there's this sort of homogeneous migration of 0.01 between subpopulations in the past, and then around 10 generations ago, the subpopulations on the left and the right experience a sudden sort of break in migration right down the middle, as we can see here in red. And this is the result of that simulated data being applied with maps. And what we're seeing is a really good recapitulation of the history. So their maps modeling for IBD tracks between 2 and 6 centimorgans, which is on average around 50 generations ago, or around 1500 years, shows that, again, that homogeneous migration between all subpopulations. But when we get to greater than 6 centimorgans, which on average is 12.5 generations, but which can be even closer, we're seeing this sort of sudden drop in migration right down the middle, which is a little bit wider, but it looks like it recapitulates the simulated data quite well. Next, they wanted to try that sort of mirror image where this imaginary world has a sort of lack of migration from the left to the right in the past. And then at some moment around 20 generations ago, suddenly all the subpopulations are migrating in the homogeneous way. And what we're seeing is that MAPS is recapitulating this result really well as well. So from 2 to 6 centimorgans, which was in the distant past, we're seeing a sort of lack of migration between populations on the right and left. And then for timing greater than 6 centimorgans, or around that sort of 12.5 generations, we're seeing this homogeneous migration. So from these migration simulations, it looks like MAPS works very well. And I noticed that these two simulations have different time points. So this one used 20 generations and the previous one used 10 generations. And I think it's worth considering why they did that. My guess is there's something that happens with the power of this tool when you get to smaller IBD tracks, but I have no idea. It's worth asking the authors. But sort of to summarize, the migration simulations look really good with MAPS. Next, they wanted to see how well it recapitulates different population sizes in different subpopulations. So in this simulation, they keep migration the same, which can be shown here on the right, where everyone is blue. And then they change the effective population size so that this middle band has a lower effective population size. And what they found with MAPS was it was able to distinguish those two different parameters. So migration stays consistent with MAPS, as we can see here. But when we look at the population size, we see this really large stretch of reduced population size. So the conclusion from these simulations is that MAPS works very well. And in the paper, they actually compared it to an existing tool called EEMS, which uses a very similar approach, but it uses genome-wide similarity, and it loses a lot of the resolution that MAPS is able to get. So it's worth digging into the paper a little bit to see just how powerful this tool is. Okay, so we've seen that with simulated data, MAPS works quite well. Now, how does it work with real genetic data? In the paper, the authors analyzed the PLoS dataset, which is a genome-wide SNP array dataset for individuals with European ancestry. And after their filtering process, which was used to remove samples with admixture, they wound up with just over 2,000 individuals. So here I'm showing the map of Europe, and this is the result when they applied their tool to this data set. And what they did was they split the IBD tracks into 3 groups, 1 to 5 centimorgans, which corresponds to around 2,500 years, 5 to 10 centimorgans, which corresponds to around 600 years ago, and greater than 10 centimorgans, which is around 200 years or more recent. And then because we're dealing with real world data, they took their migration and population size parameters and they tried to make them more continuous. So we have dispersal distance, which can be explained basically as the average distance an individual is expected to move within a generation, as well as population density, which is just population size divided by a given surface area. So the findings here are pretty cool. First, they're seeing that dispersal distance and population density are incredibly consistent across time. So what we're seeing 2,700 years ago is quite consistent to what we're seeing 675 years ago and 225 years ago. And a lot of this can be explained with different geographic barriers that exist. So most notably to me were the Alps, which are right in this region here, and we're seeing here a complete drop in dispersal distance. And that can also be seen with the English Channel and the Adriatic Sea historically. But then again, sort of also I wanted to note that some of these findings can really reveal some really cool historical empires and events. So around 2,700 years ago, we're seeing a large population density around Italy and Carthage. And I think that this is sort of my conjecture from this analysis, but it looks to me like that is that it is basically revealing the Carthaginian and the Roman empires that arose around that era. Then around 600 years ago, we see this large expansion in Normandy and a spread of those people to the Mediterranean. And in that time, we were seeing the Normans really spreading and the rise of Christendom with the recession of some of the Muslim empires of Baghdad and Cordoba. And then the last finding they observed was this huge increase in population size in the last 225 years. So these units, they didn't explain the units, but I believe it's individuals per square kilometer, if I'm not mistaken. And in the last 225 years, there's almost 100-fold increase, which we can see with the difference in these two scales. And some explanations for that are that in the last 200 years, we experienced industrialization and our agricultural revolution. So some really cool findings that historically we know about, and they can be seen in the genetic data as well, which to me is very exciting.